In this video, we'll cover the standard level core content from topic A1.2 on nucleic acids, focusing on the structure of DNA and RNA. You've probably seen DNA before. You might think it looks like the famous double helix, and that's correct, but we'll go through it in more detail. DNA stands for deoxyribonucleic acid, and it stores hereditary or genetic information that is passed from parents to offspring. DNA is a polymer, which means it's a very long molecule made up of repeating units called nucleotides. RNA is similar but slightly different. Some viruses use RNA instead of DNA to store genetic information. However, it's important to remember that viruses are not considered living organisms. All living organisms use DNA to transmit hereditary information from one generation to the next. Let's zoom in on one of these repeating units, the nucleotide. Whether we are talking about DNA or RNA, nucleotides share the same basic components: a pentose sugar, a 5-sided sugar, a phosphate group, and a nitrogenous base. The phosphate group must be attached to a specific carbon on the sugar. This detail is crucial because the attachment points determine how the nucleotides join together. The term pentose sugar is general. When we specify the type of nucleic acid, we replace it with the actual sugar name: deoxyribose for DNA and ribose for RNA. The nitrogenous base attaches to the other side of the sugar, so a single nucleotide consists of a sugar, a phosphate group, and a nitrogenous base. When multiple nucleotides join together, they form the polymer strands of DNA or RNA. To connect nucleotides, a covalent bond forms between the phosphate group of one nucleotide and the pentose sugar of the next. This bond is called a phosphodiester bond, and because it's covalent, we draw it with a solid line. This is similar to how we draw water molecule: solid lines for covalent bonds and dashed lines for hydrogen bonds. Each nucleotide's nitrogenous base can vary. In DNA, there are 4 nitrogenous bases: adenine A, thymine T, cytosine C, and guanine G. In RNA, you will find adenine, cytosine, and guanine too, but instead of thymine, RNA uses uracil, U. These bases can be categorized as either purines or pyrimidines. Adenine and guanine are purines, double ring structures, while cytosine, thymine, and uracil are pyrimidines, single ring structures. You'll often see the bases abbreviated by their first letters, but remember to write out the full names in formal answers. When you visualize DNA, you often think of its twisted ladder structure. If you untwist the double helix and look closely, you will see a sugar phosphate backbone with nitrogenous bases extending inward. The sequences of A's, T's, G's, and C's is like a biological binary code. Just as patterns of zeros and ones encode information in computers, different patterns of these bases produce different genetic traits. This sequence is what makes each organism unique. DNA is double-stranded and forms a double helix, while RNA is usually single-stranded. In DNA, the 2 strands run in parallel but opposite directions, a feature called antiparallel. The strands are connected by hydrogen bonds between complementary base pairs. Adenine always pairs with thymine, AT, via 2 hydrogen bonds, and guanine always pairs with cytosine, GC, via 3 hydrogen bonds. In RNA, adenine pairs with uracil instead of thymine. Forming a strand of DNA or RNA requires joining nucleotides through condensation reactions. This means a molecule of water, H2O, is removed when forming each phosphodiester bond. For example, when connected 5 nucleotides, you would remove 4 water molecules to create 4 phosphodiester bonds. This same principle applies in other biological molecules as well. Let's look more closely at the structure of DNA as an example. Each strand is made of nucleotides joined by phosphodiester bonds. The two strands run antiparallel to each other and are connected by hydrogen bonds between complementary bases. If you were drawing this, you should show the sugar phosphate backbone with the bases facing inward and use dashed lines for the hydrogen bonds. Remember that there are 2 hydrogen bonds between A and T and 3 between G and C. This is a great way to illustrate the full structure of a double-stranded DNA molecule. Comparing DNA and RNA highlights both similarities and differences. Both are long polymers of nucleotides and share the same basic structure: a sugar, a phosphate group, and a nitrogenous base. However, DNA is double-stranded with deoxyribose sugar, while RNA is single-stranded with ribose sugar. DNA uses thymine, whereas RNA uses uracil. In both, cytosine pairs with guanine, but the base pairing for adenine differs. It pairs with thymine in DNA and uracil in RNA. DNA structure explains how it replicates. Replication must be accurate because DNA carries genetic information passed to offspring. DNA replication is semi-conservative. Each original strand acts as template for a new strand. After replication, each new DNA molecule has one original strand and one new strand. This process relies on a complementary base pairing. For example, if the original strand reads AGGC, the new strand must read TCCG. Complementary base pairing is also basis for gene expression. Genes are expressed when the code on DNA is used to synthesize a protein. This happens in 2 main steps. First, DNA acts as a template to make RNA, then RNA is translated into a protein. Both steps depend on the rules of complementary base pairing. This topic connects to the IB biology theme of unity and diversity. The unity lies in the fact that all living organisms use DNA made up of the same 4 bases, A, T, G, and C, and follow the same rules of complementary base pairing. The diversity comes from the countless possible sequences of these bases and differences in DNA length, which result in the vast variety of life forms. DNA size and packing also contribute to its function. DNA is incredibly thin, only about 2 nanometers wide, allowing long molecules to fit inside tiny cells. Even the simplest organisms can hold billions of base pairs, storing immense genetic information. Finally, the universal nature of the genetic code is another example of biological unity. The way the genetic code on RNA is interpreted into proteins is the same for every organism, whether it's a human, a butterfly, or a tree. This strongly supports the idea that all living organisms share a common ancestor. a core concept in biology. In this lesson, we have covered 10 key understandings about nucleic acids that you need to store carefully in your memory banks. From the structure and components of DNA and RNA to the rules of complementary base pairing, phosphodiester bonding, the double helix and antiparallel nature of DNA, the differences with RNA, and the concept of the universal genetic code. Each idea builds your foundation for understanding how life stores, copies, and expresses genetic information. Make sure you revisit these points regularly as they connect directly to many other topics we will explore next. DNA and RNA are nucleic acids that store and transmit genetic information using sequences of nucleotides, each made of a sugar, phosphate group, and nitrogenous base. DNA is double-stranded and antiparallel, while RNA is usually single-stranded. Nucleotides joined by phosphodiester bonds and base sequences encode genetic traits. DNA replicates semi-conservatively, and the universal genetic code shows the unity of life, while base pair variations create its diversity. Thank you for your focus today. Keep these ideas in mind, stay curious, and I look forward to exploring more IB biology with your next lesson.